Hey everybody, my name is Jason Walker. I'm a science coordinator with the National Math and Science Initiative, and this screencast is going to be all about plant signaling. It's the last screencast in a series on botany. So let's think about plant responses to stimuli. Okay, we have to remember that unlike us, plants do not have a nervous system. So their response to stimuli is dependent upon the action of hormones. These hormones are produced in one part of the plant and they're transported to another. And what we see in this figure is a three-step process that we call a signal transduction pathway. Okay? With reception, what we have, the cell signals are detected by receptor proteins that are located in the plasma membrane. They undergo changes in shape in response to the stimulus, okay, this hormone or the stimuli. Transduction is a multi-step pathway involving relay proteins and second messenger proteins that amplify the initial signal. Now, all of this is going to culminate in a response, and the cellular response is primarily accomplished by two mechanisms: increasing or decreasing the mRNA production, which we know is made in transcription, and that's going to be sent out attached to a ribosome, and translation is going to occur where that mRNA transcript is read is used to make the polypeptide chain. Also, the other mechanism is activating existing enzyme molecules, these protein molecules that have already been made. So let's look at a real quick animation that's going to put all this together for us.
Cell-to-cell signaling, with molecules carrying signals from sending cells to receiving or target cells, is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
This animation uses an animal cell, but the process is the same in plant cells. Obviously, we do not see a plant, or I should say a cell wall here, so we know that it's not a plant cell. So what we see here are is a tropism. This is actually phototropism. Okay, tropism is the turning or bending movement of an organism or a part toward or away from an external stimulus. So this could be heat, light, or gravity. And we'll look at light gravity here in just a little bit. So I want you to pay special attention to these tropisms. They're either moving toward or away from the stimulus. If they're moving toward the stimulus, that's a positive tropism. And if they're moving away, that is a negative tropism. So when we look at these pictures right here, what is actually occurring? Okay, they are moving toward light. Okay, so that is positive phototropism. Photo meaning light. So this is positive phototropism. What's important to remember is that you're going to have to be able to explain this tropism and what's going on. But what you will not have to do is to use specific hormones like auxins, okay, that explain this. Okay, so let's look at a real quick video of the mung bean and phototropism. We'll look at positive phototropism. Now, I want you to see these leaves. These leaves are actually foraging for light. They have phytochromes. Okay, so they're foraging for light. What I want you to see is what's actually turning this plant is on the opposite side here. There's cell growth over here caused by auxin. Okay, and it's going to be on the opposite side. Same thing with these leaves of this plant. These leaves are foraging for light wavelength. Got more bluish wavelengths on the left, more reddish wavelengths on the right, and you can see that it's actually moving more toward the left. Okay, it's because of the accumulation of auxins on the non-stimulated side. Okay, causing that cell division. So some of these hormones besides auxins are cytokinins, abscisic acid, ethylene, those types of things. Again, you do not have to be able to differentiate one hormone from another. What you do need to be able to do is to list and describe some functions like stimulating cytokinesis, responsible for tropisms. They stimulate cell elongation. They slow growth. They promote seed dormancy. They promote ripening of fruit. Those types of things. So these are responses. These are hormones that elicit responses via that signal transduction pathway in these plants. Okay? So let's talk about plant responses to light specifically. Now, plants can detect light's direction, intensity, and wavelength. These light receptors are called phytochromes. Okay, two concepts that are important when we start talking about this are photoperiodism and circadian rhythms. Now, you might be familiar with circadian rhythms in your own life, as in, because at a certain time of day, your body's going to start putting out melatonin. So you're going to start getting tired at about the same time every day. You're going to wake up at about the same time every day. Just naturally, okay? Your metabolism is going to be higher at the same time every day. You're going to be more alert at the same time every day, and then we get in these rhythms in a 24-hour period. Well, these plants are going to they're going to do the exact same thing. Now, photoperiodism is a physiological reaction of organisms to the length of day or night. It occurs in both plants and animals. In the 1940s, researchers learned photoperiodism is controlled by night length. Okay, so we're talking about there's a critical dark period that these plants have to be able to be exposed to. An example of photoperiodism is when a plant doesn't bloom during the increased darkness of wintertime. The photoperiodic effects relate directly to the timing of both light and dark periods. Okay, so short day plants flower when the day lengths are less than a critical photoperiod. They cannot flower under long days or if a pulse of artificial light is shown on the plant for several minutes during the middle of the night that we see as we see in this figure here. Okay, they require a consolidated period of darkness before floral development can begin. Okay, natural nighttime light, such as moonlight or lightning, is not sufficient brightness or duration to interrupt flowering. Okay, in general, short day, these long night plants flower as days grow shorter and nights grow longer. So after June 21st in the northern hemisphere, which is during summer or fall. Note that some species must pass through a juvenile period during which they cannot be induced to flower. Now, long day plants flower when the day length exceeds their critical photoperiod. So these plants typically flower in the northern hemisphere during the late spring or early summer as days are getting longer. So in the northern hemisphere, the longest day of the year is on or about June 21st, the summer solstice. After that date, days grow shorter, nights grow longer, until the winter solstice, December 21st. This situation is reversed in the southern hemisphere. Okay, in some seasons, respectively, rather than in some parts of the world, however, winter or summer might refer to rainy or dry seasons, respectively, rather than the coolest or warmest times of the year. Okay? So, to flower or not to flower? Okay? This figure summarizes the reversible effects of red and far red light on photoperiodic response. A flash of red light shortens the dark period since it is the most effective color in interpreting the nighttime portion of the photoperiod. A subsequent flash of far red light will cancel the red flash's effect. Okay? So what I want you to do is I want you to look at this figure and let's just go through this together. Okay? Night length controls flowering and can be interrupted by a flash of red light. Okay? Flowering is controlled by the length of a critical dark period. Okay? This effect is canceled if the flash of red light is followed by a flash of far red light. Okay? So, let's go through these just one by one. Okay? Number one, the plants experience more day than night, therefore a long day plant is favored. So the long day short night plant blooms and the short day long night plant does not. Alright, let's look at what happens in number two. The plants experience less day than night, thus the critical dark period is exceeded. So the short day long night plant blooms and the long day short night plant does not. Now let's start putting in some red light. In number 3, the plants actually experience less day than night. So the short day plant should be favored, but the critical dark period is interrupted by a flash of red light, which resets the photoperiod, effectively reducing the critical dark period. Thus, the long day, short night plant blooms, while the short day, long night plant does not. Now let's see what happens at number 4. Again, the plants actually experience less day than night, but the plants experience a flash of red light followed by a flash of far red light, which cancels the effect of the red light. Thus, the short day, long night plant blooms, and the short day, long night plant does not as expected, right? Just as in number 2. Okay, well, let's look at what happens in number 5, because we're going to have red light, then far red light, and then we're going to flash some red light again. Okay, so plants experience this flash of red light. Then they experience the far red light, which cancels the first flash. But then what happens? That's followed by a final flash of red light, which activates the reset again. So the critical dark period is effectively shortened, thus the short day, long night plant does not bloom, but the long day plant does bloom, just like in number 3. Okay, now when we look at number 6, okay, the plants experience 2 sets of red followed by far red exposures. Thus the flashes effectively cancel each other out. Okay, the plant experiences a night longer than the critical dark period. Thus the short day plant blooms, the long day plant does not. Alright, let's look at geotropism. Now, geotropism or gravitropism is a plant's response to gravity. It's going to turn or move in response to the gravitational pull, that force coming down. Okay, so which way do you think a root will want to go? Toward or against gravity? And what about a stem? Which way does it want to go? Okay, well, as you can see, the seed's radicle is exhibiting a positive geotropism. Charles Darwin was one of the first to scientifically document that roots show positive gravitropism and stems show negative gravitropism. That is, roots grow in the direction of gravitational pull, downward, right? And stems are going to grow opposite gravitational pull, or upwards. So let's look at gravitropism. Okay, here's the gravitropism in the mung bean. Okay, so it's going to be turned over on its side, and you see that we actually have some light underneath. So there's 2 different stimuli. Okay, and so which is more powerful, phototropism or gravitropism? Is the plant going to grow toward the light, or is it going to exhibit this negative gravitropism and grow up? Right? What do we see going on here? Right? So, in general, gravitropism is the dominant response in roots, while phototropism is the more dominant in the shoot. Okay? In general. Now, we did see that was actually moving away from the light. That stem was, right? Let's talk about response to other stimuli, like Thigmotropism. Thigmotropism is a movement in which a plant moves or grows in response to a touch or contact stimuli, right? We see this tendril. It's wrapping around the stem as its cells elongate on the side opposite of the stimuli of touch. Next, after sensing a creature, the tentacles of the sundew perform its predatory mechanism in order to trap its prey. Afterwards, the plant digests the prey using its digestive glands. Sundews are now considered endangered due to increasing temperature as well as limited growing areas available for their kind. Finally, we see a Venus flytrap, which we talked about in the last screencast. This Venus flytrap will ensnare its prey as well. So, this is just a really cool video that I wanted to show you, and it's not very long, but you can see in these leaves, they're actually going to respond to just some minor touch. Okay, so all of these things are leaflets, all right, which we talked about. But watch this. You touch that, and look what those leaflets do. They just close up. Okay, now that is a defense mechanism. That's evolution at work right there. Now watch this. This is awesome, right? You can see that that response is going down that stem right there and eliciting that response going down. Okay, and that's just in one little leaflet. Now I just want you to see. We're going to look at the whole leaf at what's going on here. Okay, we see that these sets of leaflets are going to they're going to close up. Okay, but watch what happens. Watch what happens to these other leaves. Look at that. That response has gone to other leaflets, not on the one that was being touched, but on other leaflets. So you can see that these hormones, these responses, these can travel throughout the stem, throughout the plant, throughout the leaf. Now, if you have any questions at all, please make sure that you ask your teachers. Botany is a really exciting. They're really exciting concepts, but they can be hard to understand at times. So make sure that you consult your book, consult your teacher, and watch this screencast and the other screencast as many times. Stop them as many times as you want to. Watch them as many times as you want to. Ask as many questions as you possibly can. So when the AP Biology exam comes up, you are going to rock the botany part. Okay? Have a good day.